Morning, my name is Miss Serena Victor and I'll be talking about corals, SCTLD, and coral bleaching. So some brief history about corals is that they are 500 million years ago. So they started off as simple solitary organisms, but over the years they have evolved into the coral reefs that we know today. So they are considered animals because they don't make their own food. So they have these tiny tentacle-like arms that are used to capture food from the water and sweep into their mouths. So they get their food from algae living in their tissues or capturing and digesting their prey. So the two traumas that they are experiencing is coral bleaching and SCTLD. So coral bleaching happens when the sea temperatures get too high or too low, and that causes the algae to become stressed out. So the algae starts to leave the coral, then the coral starts to turn white, and with its main food source gone, which is the algae, it starts to become vulnerable. So the other one is SCTLD, which is also known as stony coral tissue loss disease. It's a new lethal disease that is affecting many corals such as brain, pillar, star, and etc. So this disease spreads quickly and causes a lot of corals to die. So the cause of the disease is unknown, but most biologists, marine biologists, such as Logan Williams, suspected it to be a certain type of bacteria that is transmitted into the water. And some ways to treat this disease is to use probiotics and natural ingredients or aminosylin. So they treat the whole coral and their lesions. So since corals are more risk to die from SCTLD than bleaching, our hypothesis is if reparation work is not done quickly, then most, if not all, of the corals will die, leaving humans and marine life vulnerable. Thank you. On to the materials that you would need to conduct this observation. So if you have a parent, guardian, or friend that is a certified scuba diver, they will need a wetsuit, a regulator, air tank, shin fins, the snorkels, goggles, weights for diving, a plastic clipboard, waterproof paper, and a pencil. It could be a pencil or mechanical pencil.
This experiment did not follow the generic setup of a scientific experiment. Only our scuba divers were tasked with obtaining the necessary materials and information for the experiment, which were given to us to create this lab video. Good afternoon, my name is Erna Christopher, and I will be talking about the claim, evidence, and reasoning of this observation. Our claim is that the stony coral tissue loss disease occurs more rapidly than coral bleaching. It will also continue to affect local coral reefs unless treated efficiently. Our evidence is supported by the graph below. In the areas OFAV, MICAV, and SSID held diseased corals. These areas were quite far away from each other but resulted in more cases than bleaching, which was only spotted in the OFRA section. Now for reasoning. When SCTLD is present on a reef, it spreads quickly. The bacterial pathogen is transmitted via direct contact or through water columns. This was also discussed with Logan on the field trip. Unlike bleaching that is seasonal, SCTLD occurs all throughout the year. As summer approaches, it will occur more quickly and harm or kill all surrounding corals.
There's a small slideshow of the different pictures that I took when we were on the trip, starting with the corals that were affected by the SCTLD disease. You can see that it's mostly bleached. Next, you can see the starlet coral that was affected by the dark spots disease. And here, the same thing. These are the corals that we saw that were affected by the SCTLD disease. The SSID, or the starlet coral, was the only coral to be affected by the dark spots disease and was most affected by the Skittles disease as well. Shown here.
Okay, the reason that I chose this graph is because Excel wasn't really cooperating with me, so I couldn't really, the histograms didn't really look right, so I didn't use that. It didn't have an error bar suggestion or option. So basically, I just picked this graph because it was easier to read and from all the data that we collected. So on Excel, it's called either a cluster column, and it basically have all of the data that we collected. Like it was color coded, so blue is healthy, orange is like bleaching, gray is SCTLD, and like the yellow is like DSD. So it basically just like color coded everything, and that's why it was so easier to understand because it was color coded. And yeah, that's why I chose this graph.
Okay, I'm with Candice with Franklin, one of our scuba divers now collecting my data. So Candice, how was it?
It was good. When you're underwater, it's kind of like a different world. So it was nice to see all the fish and the coral that remains at coral.
Okay, did you see any bleaching?
Not as much bleaching, more of the Skittles, because bleaching is not around this time. As summer goes on, when the water gets hotter, we'll see more bleaching. Hopefully not, but it's expected. Thank you, Candice. Welcome.
In conclusion, we have realized the emergence of coral situations surrounding the Virgin Islands. Coral reefs have become more susceptible to disease as the climate change and ocean acidification levels increase. This experiment has also shown how the disease can move to other corals that are not even next to each other. The SCTLD has affected many corals in our waters and will continue to do so unless there is legislative and scientific action taken. With more research, scientists may be able to pinpoint the bacterial pathogen that has created this monstrous disease. Legislative action will also include informing the community and protection of coast while scientists treat these corals. These suggestions will increase community involvement, scientific research, and healthy coral populations. We do also believe that young people should become more involved in this recovery project. Education and on-hand learning will give students the knowledge to take action in coral recovery. This bibliography portion holds a list of websites that we use to refer back to our experiment of the Skittle disease, as well as coral bleaching and corals in general. The first bullet is by Virgin Islands EPSCoR, which covers stony coral tissue loss disease. It basically speaks about what the disease is and how it involves the Virgin Islands, which is what we focused on since we live in the Virgin Islands. The second bullet is from the U.S. Department of Commerce, which focuses on National Oceanic and Atmospheric Administration. And basically the question of this article says, what is coral bleaching? So it explains what coral bleaching is, how it affects the corals, and what we can do. And the third bullet is the Ocean Portal team, and this title is Corals and Coral Reefs. Basically, it talks about what corals are, how they affect man.
The reason why a lab video is important to do is because it's basically like you format all your words to like tell people what is going on in your lab. So it helps you, like, basically it's like public speaking, but it's not really public speaking, but it helps you like speak more clearly so people can hear you. And yeah, and you could like do all kind of editing videos to make your video look better, and it gives you more practice for future projects. So yeah.
The benefits of creating a video lab report is that it helps you keep visual document of what you have or have not done and or experienced, and if you need to go back on it, then you can.
I believe that lab videos are most beneficial for all types of experiments. They help viewers understand what is being taught in a video format. Visual representations along with verbalization smoothly connects the most important components of an experiment. For all types of learners, it is a blessing, as they can see for visual learners, hear for auditory learners, and perform similar experiments for kinesthetic learners.
She a little blurry.